Today we will discuss the amino acid transcription and translation. You see the central dogma, that is from DNA to RNA and RNA to proteins, and DNA to DNA is the replication. So, in the central dogma mean from molecular basis of life as the DNA, DNA to transcription is the mRNA, mRNA to protein. So, the amino acid is a very important, or it is a monomeric unit of the proteins. So, we will discuss little bit amino acid, then transcription and translation. So, the amino acids we contain near about nature contain near about 300 amino acids is known so far, and out of these 20 amino acids are usually it is used in the human being in the different protein molecule synthesis, and this out of these 20 amino acids or 21 amino acids, and there are 10 amino acids, that is the 9 are the essential, one is the semi essential amino acid, that is these amino acids are essential amino acids are those amino acids, they are essential to body, but their body cannot synthesize, it has to be supplemented from others, and these essential amino acids are very important for the protein synthesis. So, coming to the DNA, DNA is it is present in the nucleus, they are the monomeric unit contain the nucleotides, and this all these nucleotides, that is the DNA, near about 600 crores nucleotides present in a human being. So, they are packed in a 22 autologous chromosome and one pair of sex chromosome, and they are tightly packed and remain inside the nucleus. So, this is the DNA is the double helical structure, which is given by the Watson and Crick model. The double helical, they are double 2 molecule remaining 5 prime to 3 prime and 3 prime to 5 prime, that is the anti-parallel, they coil each other. DNA function is carry the blueprint for the life, that is the inheritance, this it contain the gene, and the gene carry the information. The gene structures, that is gene must have the exon, start site and the control site. Then DNA, as I told in the central dogma, it is a transcribed to mRNA. So, mRNA where the triplet code is there, and this tRNA contain the clover leaf structure, it contain the anticodon, and ribosomal RNA it acts as a platform where protein synthesis occur. So, mRNA after transcription from DNA, when it is transcribed, so it is known as heterologous mRNA. So, this has to be the intron and exons are there are some group which are not transcribed. So, the intron has to be spliced out. So, mRNA, for a matured mRNA, this post transcriptional modifications occur, the splicing again is 5 prime methyl guanosine capping and poly A in the 3' polyadenylation. So, these are the important events occur in the mRNA. So, that this 5 prime and the methyl guanosine capping it stabilize, and also in the 3 prime and the polyadenylation part, where also it prevent the exonuclease activities. So, that the mRNA stability is there, and after splicing out the matured mRNA is synthesized. This is splicing, as I told, this exon that example, the exon 1, 2, 3, in between the intron 1 and 2, 3, they have to be spliced out and the matured mRNA is synthesized. The translation in the central dogma, I am repeatedly telling that DNA to transcribe to mRNA and mRNA to the proteins is the translation. Translation is the simple literal meaning, that means one form of alphabet change to the another form. It is commonly like you translate from Hindi to English, that Hindi one alphabet, English another alphabet. So, similarly, in the genetic code, this mRNA content is genetic code which translated to a amino acid monomeric unit. Here is the nucleotide monomeric unit, which is translated to the protein which contain the amino acid is the monomeric unit. So, it is known as translation, that is from mRNA to protein synthesis, the process is known as translation. So, the protein is just a basic information. The proteins are the larger molecules, they are help in the structure, formation of the structure of the cell or a tissue, or it is as a enzymatic fraction, all practical purpose, all the enzymes are the proteins, also exception is the some RNAs, and they are the receptors, they function as a hormone, they act as a transporter, they act as a immunomodulator, a immunoprotein. So, the protein is a essential component of a living system. Protein structure is the amino acid is the monomeric unit, they are simple, the simple peptide chain is the primary structure, then it is a helical, the beta pleated sheet, and the 3 dimensional structure, the quaternary structure, primary, secondary, tertiary, and the quaternary structure. So, gene expression that or the regulation of the genes regulation, that is from DNA transcription 1 regulation, from transcription to translation is the regulation. So, the expression of the gene, that is from DNA to protein synthesis, they are regulated fashions, they are very strictly regulated. All the stages of these events are, they are complex and they are regulated. Like for example, because DNA is there in the nucleus, and from the protein synthesis occur in cytosol. So, you see the transcription occur, then it is come to the cytosol, where the ribosome is there and the protein synthesis will be there in cytosol. So, you see from transportation from nucleus to cytosol, it is not a simple event, it is a very regulated event. Whenever necessary, that will be transcribed and it will be transported to the cytosol in the particular mRNA platform. So, gene expression regulations are very synchronized, coordinated, it is a very complex process in a living system. The molecular biology, why we read this transcription, translation, and how it help us? You see, one, if you know a particular things, then in a modern medicines it help to develop the personalized medicine. Mean, you see particular medicines react to a particular individual in a particular fashion, you cannot generalize. So, it depends on their molecular structure, this genetic pattern. So, they are very important. So, knowledge of this molecular knowledge helping the medical profession for particular target specific, disease specific, tissue specific therapy. Another is the gene therapy. So, all the genetic disorders can be altered in the gene and the therapy can be done. Then comparative genomics, that is this knowledge, the molecular knowledge helps for this. If you comparative genomics, you can develop the molecules for the therapeutic purpose. The central dogma I am repeatedly telling, this is the from DNA to mRNA and the protein synthesis. So, the storing of the informations is there in the DNA. DNA is it stored and it transmit the genetic information. The proteins are a chain like structure, is already I told, they contain a monomeric unit is the amino acids. DNA, it contains the nucleotides, that is the purine and pyrimidine, the A and T, G and C, 4 type of different nucleotide in the DNA. At same time in the RNA, instead of thymine, it is the uracil, AU. Protein structure, I already told, it is the primary structure, helical structure, the beta pleated or alpha helix, and the secondary structure, these are known as secondary structure, tertiary structure and quaternary structure. Already I told these are these all the diagrammatic representation. This is the quaternary structure, different proteins in a 4 dimensional. This is hemoglobin is a example of the quaternary structure. So, if you will summarize, protein are the polymer of the amino acid linked through a peptide bond, and they are very specific. If you will change this specificity, the sequencing, that alteration of the proteins occur. The bright example is the sickle cell anemia. The glutamic acid is replaced in the 6th position with glutamic acid is replaced with the valine. Glutamic acid is the acidic amino acid replaced with the neutral amino acid. The overall charge is disturbed. So, they are very sequence and specific. You cannot haphazardly put the amino acids, they are very specific. The function, that is they provide structure, they carrying transport, they are enzymes, they are hormone, they are immunological protein also. Genes and protein, previously it was things usually one enzyme, one protein and one genes, that is the one gene specific for one protein. This one gene, one polypeptide also sometime, it is because protein is a polypeptide, means monomeric unit is the amino acids linked with the peptide bond. So, it is polypeptide, more than one peptide bond is the polypeptide. So, one gene, one polypeptides. So, it carries information, the DNA to transcription and transcription to translation. So, the information from DNA is their inheritance from generation to generation. So, transcription, this is the some things I will tell about. The transcription is the mRNA synthesis. So, this DNA to RNA, the transcription, the mRNA synthesis. So, where the basic unit of the mRNA or a RNA is the A paired with the uracil instead of T, thymine. So, for a protein transcription, RNA polymerase is necessary. You can ask one question, whether DNA is first synthesized in nature or RNA is synthesized first in the nature. So, that you can see that the anda or murgi wali questions, whether that hen is first or egg is first, like this, whether DNA is first or RNA is first, but evolutionary the RNA is synthesized first. It is this usual belief. What you see, the RNA polymerase can synthesize or polymerize de novo. The polymerization can start in a de novo synthesis occurs in the RNA, but DNA polymerase, for polymerization RNA polymerase is needed, in the DNA polymerase is needed. For a DNA polymerase basic function, it need 3' hydroxyl and free, but in RNA it can de novo synthesize. So, that is why it is told the RNA transcription is or RNA synthesis first occur. So, this transcription follows same base pairing rules, that is DNA replication, as but only the U is instead of T it is U, and in the RNA transcription RNA polymerase is a essential component. So, one DNA strand is there, because it is template directed, it will take information from the DNA, then one RNA polymerase enzyme is essential, then all the 4 nucleotide, that is the adenine, uracil, guanine and cytosine. All these the 4 2 purine and pyrimidine nucleotides is essential requirement for a transcription. The synthesis of RNA is from prime to 3' direction. It is a template directed enzyme, that is the RNA polymerase. It take information from the DNA to the RNA. The complementary base polymerizations occur, it take the RNA polymerase. RNA polymerase usually has got different subunit, alpha, beta, beta dash and the sigma subunit. Alpha, beta and beta dash subunit, this is a core enzyme, and sigma units, it recognize the initiation site after the promoter site recognition it binds that and the polymerizations occur complementary to the DNA. Prior to that DNA double helix has to be it made single stranded and the polymerizations occur in the 5 prime to 3 prime direction. So, the process is the initiation, elongation and termination. So, this sigma factor recognize the promoter site where the RNA polymerase binds and the initiation of transcription occur, then the elongation according complementary to the nucleotides in the DNA its complementary bases it reads and the elongations occur till the termination signals come. This hairpin looping and poly U point that is the termination site, and when the termination informations occur it the termination the transcription process ceases. Next the DNA replication. This is one thing I will apply DNA replication, that is the DNA to DNA synthesis. It replicate is the semi conservative type of replications occur, that one DNA strand that parent strand to a nascent strand synthesize, another is the one parent strand acts as a platform and the nascent strands the DNA synthesis occur. So, it occur from 5 prime to 3 prime direction, always direction is fixed. So, in the DNA replication one is a continuous segment, another is the Okazaki segments. Then after that this polymerization the DNA ligase join and nascent DNA synthesize and the new DNA is formed. It is a semi conservative type of replication, mean one parent strand, one newly the nascent strand, like this it contains the semi conservative type of DNA replications occur. The termination of the both in the DNA replication and RNA polymerization this occur. In the DNA replication there is a basic difference that the DNA polymerase has got a very specific multifunction enzyme. They have got a 5 prime 3 prime exonuclease activity, that is remove the primer, and 3 prime 5 prime exonucleate activities, that means is a editing function. That editing function is very specific, very active in the DNA polymerase. That is why the if you compare the RNA polymerization, the mutation rate is 10 to the power minus 5, here it is 10 to the power minus 3. It is very less in comparison to the RNA polymerization, because here the nature cannot afford to have a incorporation of the mismatched nucleotides. RNA because unstable very quick, but DNA is very stable, it years together it remain and it carries the genetic information. If you will summarize the transcription, that initiation, elongation, termination both in DNA polymerase also occur same thing, and it require a template and one enzyme is the RNA polymerase. Now, come to the translation part. Translation is the protein synthesis. The protein synthesis, that is mRNA act as a platform. In the prokaryote and the eukaryote, the ribosome which act at the base where the mRNA remain there, the ribosome act as a platform, mRNA provide this genetic code and tRNA is the adapter molecule which brings the activated amino acids. So, in the prokaryote it is the 50s and 30s subunits, and usually eukaryote is the 60 to 60s and the 40s subunits. So, here you see according to the genetic code, the mRNA, that is the tRNA, brings a specific amino acids. We have that 64 genetic code. See, this is the 64 genetic code UCAG, UCAG. Like this you can arrange all the 64 genetic code, it is almost universal. Now, it is known as semi universal, because some genetic code where termination codon and initiation code is altered in a mitochondrial DNA. After the knowledge of this mitochondrial DNA, this genetic code is known as a semi-universal, but it is almost universal mean it is there in prokaryote, eukaryote, plant, everywhere the same type of genetic code is there. This genetic code present in the mRNA. Ribosomal RNA acts as a platform where protein synthesis occur, that is in the they have got two subunits joined together, that is 50s and 30s. In the eukaryote it is 60 to 40s. So, ribosomal RNA I told this, that is the different subunit according to a sedimentation coefficient, they are grouped accordingly. So, that is the translation adapter molecule, that is the tRNA. So, tRNA brings the amino acids, the site of the protein synthesis. This is known as translation adapter molecule. They are clover leaf appearance. You see, clover leaf 5 prime and 3 prime, 3 prime here the amino acids are charged, and one is the D loop, it is the anticodon loop, and the another loop is the T5 loop. So, where the anticodon reads with the mRNA codon, and the specific amino acid signals come and they bind the specific amino acids with the help of tRNA, amino acid tRNA synthetase enzyme. It needs that the ATP energy driven to activate this tRNA. Codon anticodon association mRNA, this is the one picture shown how the anticodon reads with the codon of mRNA. So, important is tRNA, mRNA is the code, triplet code, anticodon association and the specific amino acid they will bring. This is the summary of the initiation. So, initiation of protein synthesis, that the if you will take for example, the prokaryote, if you will take that 50s subunits, the 50s and 30s subunits, what happened? They remain in association, they have got the initiation factor IF1, 2 and 3. So, what happened? This IF1 and IF3, the initiation factor 3 helps in the initiation codon in the mRNA, and the mRNA AUG is the usual initiation codon. So, it is recognized and initiation factor 1 help in the recognition. So, once the initiation factor binds there, so the 50s subunits dissociate and it recognize the search for, it is a random searching pattern in mRNA from 5 prime to 3 prime direction. It search whenever this initiation codon it stick there, and the according to the initiation codon, the anticodon, the tRNA will bring the activated tRNA to site of the protein synthesis. So, it required the initiation factor IF1, IF2 and IF3. IF2 help to brings the formulated methionine in the that charge with the tRNA to site of the protein synthesis. Elongation then in the protein synthesis ribosome, that is the P site and the A site. In the P site, the binds the formulated methionine find the initiation codon there, and the tRNA bind with the formulated methionine. Then what happens? The elongations occur, the next peptide bond has to be synthesized. So, it is helped by the elongation factor Tu. So, elongation factor Tu, it brings the next amino acids charged with the tRNA according to the anticodon. They are very specific, it cannot or it cannot afford to have the improper amino acid. That is why this tRNA synthesis is very specific. There are almost 20 types of tRNA synthetase enzymes are there. So, one enzyme, one tRNA enzyme for the one amino acid. So, once the improper amino acid will be there, it will lead to the hydrolytic site, because tRNA synthetase has got a acyl site and peptidyl site. Once improper amino acid will be there, they will die. So, the elongation factor T it brings this tRNA in the A side, and then peptide formation will be there, and there will be translocation. So, like this the elongation of the peptide chains occur. This is the diagrammatic representation how the elongation factor Tu brings the activated next the tRNA charged with the amino acids, and peptide forms is synthesized peptide bond है ना, P side the A side, then it translocate to the peptide with the help of peptidyl transferase from P side to A side, then again the translocations occur, then P side to A side, like this a continuous chain will be there till the termination codons will be there. Once the termination codon will be there, then what happened? The P site will remain open, there will be no amino acids tRNA containing peptide chain. So, immediately the termination factor will occupy there, and the peptide that tRNA will dislodge, and the peptide that tRNA will be hydrolyzed containing the nascent protein peptide chain. So, then it will be dislodged, and the total after this newly synthesized peptide chain will undergo the post translational modification, and after the post translational modification protein become the physiologically active protein. So, to summarize, the proteins is synthesized in the process of translation. After translation, the post translational modifications occur. There usually covalent modifications, either sulfation, phosphorylation, dephosphorylation, they become the physiologically active protein, and the proteins start their functioning. So, these are the things I told. The terminations occur, the after this termination codon is there UAG, UAA, UGA. The replication part I told is the DNA for a information, the DNA to DNA synthesis known as replication with the help of the DNA polymerase, that needs DNA template, then the nucleotides and the enzymes DNA polymerase. The type of replication is the semi conservative types of replication, usually there are also conservative type of replication. Thank you very much.